Good afternoon, everyone, and welcome to the PNI Teatime Webinar Series. If you've attended these series before, welcome back. And if you're new, thank you for attending. So today we've got a talk from Professor Dagmar Fischer of Friedrich-Schiller University in Jena. And Professor Fischer will be discussing breathing new life into old antibiotics, bacterial biofilms as a challenge for nanoparticles in cystic fibrosis. Just a couple of other webinars that we have coming up. So on the 26th of May, we're doing a joint webinar with the Control Lead Society of the Benelux and French chapter, and we'll have our very own Dr. Samuel Clark presenting and Peter Varder from University of Utrecht as well. So the discussion will be mainly around mRNA and RNA delivery therapeutics. And then for our next Tea Time webinar on the 3rd of June, we'll have Blessing Iluchonu from University of Utrecht, and she'll be discussing her work on designing nanogels that can easily be injected into vitreous cavities using small needles. So on that note, I'll hand it over to Professor Fischer.
So hello, everybody, and good afternoon. Thank you very much for your kind introduction, and also thank you for giving me the opportunity to present some of our data here in this very interesting setting. And I learned that this is a good chance to reach people from all over the world and also from very different topics. I'm going to talk about breathing new lives in old antibiotics, and I would like to answer the question how to overcome bacterial biofilms and also bacterial resistance mechanisms by the use of nanoparticles in cystic fibrosis. And let me start with some facts about cystic fibrosis as a short introduction. In old textbooks, we can find a sentence about cystic fibrosis, meaning, woe to the child who tastes salty from a kiss on the brow, for he is cursed and soon must die. And this is exactly what I'm going to talk about in the next few minutes. And the former times, the people were quite right. Cystic fibrosis is the most frequent life-shortening genetic disease within the Caucasian population, with a frequency about 1 case in 2,500 live births. And only about 50% of the diseased people reach an age of 18 and older, so this is a quite low number. It's an autosomal recessive disorder of the so-called CFTR gene, and this is the cystic fibrosis transmembrane conductance regulator gene. Meanwhile, we know about more than 1,000 mutations of the genes, and this is a quite large number in different areas of the gene. The CFTR functions as a chloride channel in the apical membranes of epithelial cells, for example, in exocrine glands and secretory cells, and therefore it is involved in the production of sweat, digestive fluid, and mucus. And this brings me to the many, many different pathophysiological changes in different organs, not only in the secretory cells, but also all the other cells and organs expressing CFTR. For example, the lungs, the pancreas, the liver, the reproductive tract, the stomach, and so on. The most striking changes are seen in the airways, and this is what we are interested in the treatment of cystic fibrosis. Typically, the impaired transport of chloride iron results in the production of a highly viscous mucus, which clog the airways due to mucus accumulation and also decrease the mucociliary clearance. And this is connected especially in chronic diseases, or in this kind of chronic disease with inflammation and also with the chronic lung infections. Over 80% of the people that are older than 18 suffer from a chronic lung infection, and this is caused by Staphylococcus aureus, Pseudomonas aeruginosa, and Haemophilus influenza, the most important microorganism in this kind of infections. And approximately 90% of the patients die from the lung destructions promoted by this kind of pathogens. And conclusively, the antibiotic therapy and treatment is a cornerstone in the CF treatment to prevent the chronic diseases, to reduce the bacterial load, and also to restore the pulmonary functions. And typically, 3 different antibiotics are used in the late stage of the disease, like colistin, tobramycin, and aztreonam. And they are administered into the lung as a powder inhaler, as a nebulizer, means by inhalation to achieve high pulmonary concentrations and also to reduce systemic side effects. However, we have a lot of challenges for inhaled antibiotics to treat pulmonary infections. One of my PhD students always called it is a little bit like a James Bond, or has the job of a James Bond. And after the successful inhalation to the deeper lungs and also the deposition on the mucus, we need a dissolution of the antibiotic in the mucus, followed by a diffusion through the mucus, and followed by a penetration into the biofilm to kill the bacteria. But many antibiotics are unable to really reach the pathogens because they interact with some components of the mucus, for example, by electrostatic interactions, or they demonstrate a limited diffusion into the high viscous mucus. They have a rapid clearance by the lung, for example, by the mucociliaris activity, or also by macrophages. And if they reach the biofilms, they sometimes interact with resting bacteria without any kind of metabolic activity. So a lot of hurdles that have to be overcome. We were interested in tobramycin, an aminoglycoside for the treatment of cystic fibrosis, especially gram-negative bacteria. And in late-stage diseases, the tobramycin is no longer able to penetrate the mucus, the biofilm, and also to interact with the bacteria. This means it has a low antibiotic effect. This is mostly due to some bacterial resistance mechanisms. We decided to go for a nanoparticle as an antibiotic drug delivery system, and we use polylactide co-glycolide, which is a well-known biodegradable polyester already known from many applications on the market. And we used it as in the combination with the covalent binding of polyethylene glycol, which means in the abbreviation PEG, which should act as a stealth polymer to inhibit interaction with or unspecific interaction with components of the mucus. And to be able to track the particle in our biological settings, we used AMCA, which is a coumarin derivative, the amino-methyl coumarin acetic acid, to blue fluorescently label the particles. Let me start with an introduction into the preparation of the tobramycin-loaded particles. Tobramycin is a highly hydrophilic polymer drug with a log P value of 5.8, and it was incorporated into the PLG by double emulsion evaporation method. We used AMCA PLGA and pegylated PLGA in a mixture of 2 to 3. And formed a water and oil emulsion by dissolution of the polymer in acetic acid as organic phase and the tobramycin in water as aqueous phase. And the homogenization was performed by ultrasound. Afterwards, we poured the water and oil emulsion into a second aqueous phase containing 2% PBA as a stabilizing agent, and formed a water and oil and water emulsion by ultra-Turax, followed by evaporation of the solvent, followed by centrifugation and collecting of the nanoparticles and lyophilization. We prepared two different types of nanoparticles with about 200 nanometers in size and 900 nanometers. The hydrodynamic diameter was measured by laser light scattering, and the polydispersity was quite low, below 0.2. The tobramycin loading did not change the particle size. And this was different for the zeta potential. The blank particles demonstrated zeta potentials of about minus 30 millivolts due to the acidic character. And the incorporation of tobramycin reduced the zeta potential down to minus 10 to minus 15 millivolts due to the cationic charge of the tobramycin masking the anionic acidic charges. The particle size was confirmed by scanning electron microscopy. We found also spherical particles with a smooth surface, and the drug load determined by HPLC measurements was comparable for both type of particles, around 1.5 to 1.7 micrograms per milligrams. To be able to use these kind of particles in vitro and in vivo biological experiments, we need a high biocompatibility. And this was checked in the human epithelial cell line A549, treating the cells over 24 hours with different nanoparticles and quantifying the activity, the cell viability by a cell theta glow luminescence assay. Therefore, we used the quantification of ATP to detect the viable cells. Independent from the concentration up to 1 milligram per milliliter, independent from the particle size, and also independent from the presence of tobramycins, all particles were found to be absolutely viable up to 100%. And we did another experiment. We pre-incubated the nanoparticles with the artificial mucus and checked again the cell viability, and the cell viability again was high. It was in this case a little bit higher than 100% because the mucus acted as a nutrient for the cells. If you talk in the following about artificial mucus, we have a mucus consisting of mucin, DNA, casein, some salts, EDTA, and at the end we add egg-yolk emulsion to have a kind of lipophilic part. And this artificial mucus is not really a real mucus, but it enabled us to work on the standardized conditions. Biocompatibility is one part. The other part was the question whether the particles are able to permeate the artificial mucus, as we identified this at the beginning as one of the challenges for the particles. And therefore, we used a two-chamber system called inserts with 8 micrometer pore size. In the upper chamber, the artificial mucus was loaded with AMCA-labeled particles, and the time-dependent transport of the particles through the mucus into a second basolateral compartment, simulated lung fluid, was investigated and quantified. And we found two typical effects when we checked the permeated particles over time. On the one hand, the smaller nanoparticles penetrated the mucus pretty much better than the larger particles. I mean, I think this is pretty easy to explain. And on the other hand, the tobramycin containing particles passed the mucus more efficiently than the drug-free particles. And this can be explained by the different zeta potentials. The higher zeta potential of the drug-free particles enabled more electrostatic interactions with the mucus and therefore inhibited the penetration. But the most important experiment was the biofilm experiment to investigate whether the particles are effective against microorganisms or not. And these biofilms were performed under static and dynamic conditions in artificial mucus. For the static conditions, we used bacteria grown in an artificial mucus containing jumbo slides, and we followed the penetration of the fluorescently labeled nanoparticles by confocal laser scanning microscopy. And on the other hand, we stained the bacteria with a live dead kit. I will get back to this in 1 minute. On the other hand, we used a dynamic in-bioflux chamber, which is a microfluidic device imitating the shear flow in the human body, to also get an impression of the influence of the shear flow for our experiments. Let me start with the static biofilm experiments. I brought you two examples, the Pseudomonas aeruginosa and the Bacteria cepacia experiments. If a patient is infected with Bacteria cepacia, then this comes along with a very decreased survival, typically over up to 6 months. You can see the control cells in the phosphate buffer cell line, the biofilm formation, and all the time when you see green, this means we used the live dead kit staining, and this means viable cells. It's like a traffic light code. If you see red, then the bacteria were killed. Tobramycin up to concentrations of 1,000 microgram per milliliter did not work. You see again the green. Also the free nanoparticles here, the larger one, did not work. And the blank mixture, a physical mixture of nanoparticles and tobramycins were also not effective. But the picture dramatically changed when we used the nanoparticles with the incorporated tobramycin. Independent of the particle size, all the particles were able to work. You see now the red color, which means we were able to kill the bacteria. And you can also see that we were able to disrupt the biofilm, especially seen here for the Pseudomonas aeruginosa. This means, in conclusion, that the particles obviously were able to penetrate the mucus and the biofilm and show the superior efficacy compared to the free drug. We are now under investigation whether this is not only the biofilm penetration, but maybe also the overcoming of a bacterial resistance mechanism. This was the static experiment, but what happened under fluidic condition in the microfluidic setting? And here we have exactly the same phenomenon. Again, the control, the tobramycin, the free nanoparticles did not work, but the tobramycin encapsulated in the nanoparticles showed superior effects, even in the more physiological environment. And it's important to state that we used here 0.8 micrograms tobramycin per milliliter, which means the concentration of tobramycin which is necessary is significantly lower. However, you cannot inhale nanoparticles. Typically, they are inhaled and immediately exhaled in the airstream, and therefore we decided to produce larger particles because for the deposition in the deeper lung area, 2 to 5 micrometer particles are preferred. And we used for this a vibrating mesh nebulizer, the IH50 from Boira, to produce aqueous droplets in sodium chloride containing our nanoparticles. And I would like to focus you on the MMAD, which is the mean aerodynamic diameter. It was for our nanoparticles below slightly over 5 micrometers, and the fine particle fraction below or smaller than 5 micrometers was about 50%. This means very good conditions for our experiments. And the output rate was not very changed compared to the cell line. It was about 500 micrograms per minute, which means we should have sufficient amounts for biological effect. So in conclusion, it's possible to produce by nebulization droplets that should be efficiently deposited in the deeper lung. But how to test this in a physiological setting? Therefore, we used a Challis-Hens egg model. This means we used fertilized eggs from a local supplier, and the eggs were, after a pre-incubation time of 72 hours, transferred into a petri dish into a Ringer solution. Afterwards, we transfected the eggs by Pseudomonas aeruginosa, which was transported or deposited in a ring system, and we checked the distribution of the bacteria by their fluorescence using the so-called molecular light system. And compared to the negative control only in media, you can clearly see over time that there is a growth of the bacteria, and the bacteria killed already after 10 to 24 hours all the eggs. To be able to simulate this, the aerosol deposition, we constructed a device for the application of the aerosol by a 3D printer. It's a plastic device which was validated and makes us pretty sure that we have a certain concentration applied on the eggs. So what happened after application? I would like to show you the data as survival over time. So we applied the materials and checked the survival over time, and each plateau represents the number of killed eggs. First of all, I would like to focus you on the non-treated eggs, only treated with Pseudomonas aeruginosa, and most of the eggs were already killed after 16 to 40 hours. This is in huge contrast to our nanoparticles, here the blue curve. We use PLGA particles and pegylated PLGA particles containing tobramycin, and we have here up to 15 hours, more than 50% survival. This means that the system worked again. When we check the concentration of these particles, we have about 12.5 micrograms tobramycin, which itself, this is the green curve here, were not very effective. To be effective, we had to use free tobramycin in a concentration of 5 milligrams. This means a pretty much higher concentration. Consequently, we have here exactly the same effect, that obviously the nanoparticle-based systems worked pretty much better in these kind of systems compared to the free tobramycin. What is next? It means, what did we learn from these experiments? We learned that it is really necessary that we have larger particles for the lung application. At the moment, we used aerosol droplets, which are now under investigation in an animal experiment. But we also want to use for further experiments so-called blend systems. This means we would cover the surface of larger carrier microparticles made of lactose or mannitol with our nanoparticles, or alternatively nanocomposites microparticles where the single particles stick together to larger aggregates. On the other hand, we learned that for our preparation of the particles in an industrial setting, it would be nice to have an organic solvent-free system. Typically, we use ethyl acetate or dichloromethane, and we now started to use so-called green methods or sustainable methods, where we replace the ethyl acetate, the organic solvent, by sustainable green solvents like polyethylene glycol of low molar mass, or styrene, which is a glucosinone derivative made of cellulose, or different other systems. This means alternatives to organic solvents are a huge topic at the moment in this field of particle preparation. Let me conclude my data. I showed you that we have a very nice penetrating system for the antibiotic delivery with a high in vitro biocompatibility. We are able to kill the antibiotics in the biofilms with a high efficacy and with a low tobramycin content. We can nebulize the particle to deliver them efficiently into the deeper areas of the lung. And we are looking at the moment how we can apply these techniques to other research fields, for example, the transfection of DNA or RNA, or the encapsulation also of natural substances for anti-inflammatory activities. I would like to thank the people of my lab who did the work, especially Lisa Müller-Bötticher and Julia Ernst, and also Katrin Arnold. And a special thanks goes also to the Institute of Infectious Diseases and Infection Control of the clinics here in Jena, the lab of Matthias Pletz. I would also like to thank all the people who gave the money, and I would like to thank you for your attention, and I'm really happy to get into discussion with you and take your questions. Thank you very much.
Thank you, Professor Fischer, for a very, very interesting talk. And it's good to see that with the use of the aerosol, you can see there's some clear therapeutic applications as well. So that's good to see. Thank you, Professor Fischer. Thank you, Richard. Thanks, everyone, for attending. This has been recorded as well, so we can share the recording with you if you'd like to view it again. And if you have any questions, please feel free to contact us. I'd also like to introduce Martin Rabel and Jürgen Schotig, who are both based in Germany as well. So please do feel free to contact them. And also, as I mentioned at the start, we do have a couple of other webinars coming up. So we have Peter Varde from Utrecht, who will be presenting on the 26th of May in conjunction with the Controlled Release Society webinar that we have. And then we also have Dr. Samuel Clark from Precision Nanosystems, and they'll be discussing the scalability of mRNA lipid therapeutics. And then for our next Teatime webinar on the 3rd of June, we have Blessing from Utrecht as well, who'll be talking about nanogels. So all these links will be sent out on our social media as well. So please feel free to join us on LinkedIn and Twitter, and also on our website to register for these events. Thank you very much, and we hope to see you soon. Bye-bye.